Ooh. Yay. Let's see. So off with. Oh, it's so weird to look at. I went over that today. I hope I did this right. If I didn't do this right, I'm gonna be so sad. Okay, let's. Still the same thing. Okay, I hope that's correct. When I was going over measures of sexual tendency. I wonder. Oh yeah. Emails, sent out emails. Oh god. Are you gonna sneeze? Oh my god. Ow. That was embarrassing. Huh? Oh, okay. I already have. I have a theory. I know, I'm not studying right now at all. I'll be back in like a few minutes. Give me a sec. Okay, I'm back. Where's the invitation? Oh, this isn't due till Friday. Yes. I just remembered. I thought this was due. Okay, let me finish. It's gonna be due 15th. I'm just gonna put this day, or Thursday. Yeah. So I don't need to finish this now. What else will I work on? I need to work on my honors homework, because I think that's due tomorrow. Yeah. Yeah, that one's due tomorrow at 8 a.m. I think I remember. Yeah, the research one. Okay, I'm gonna work on that today. And yeah, it's due tomorrow. So I'm gonna try to finish it today, turn it in today. Because they like to see you turn in stuff early. Down it. That is weird. That's weird. I don't even think I have any photos of myself on here. Um, that's really weird. Alone. Okay, I'm gonna save this for another day. Save selected. Rid of that. And I'm gonna work on my honors homework. selected the other professor. Mhm. Okay. The fact that she got her bachelor's in zoology of all things is just it's beautiful. Chemistry. Like she got 3 bachelors. Chemistry, zoology, microbiology. That's wild. Some of these professors, man. I'm just like, who are you? Chemistry. If I can type. Oh my god. PhD. And then she finished with cognitive neuroscience. Oh my god. So cool! I want to meet them. Receive. Competitive funding. And teacher assistant mailing. She's worked a lot in Maryland. Divine Brain Bee. Project Synapse. Oh. I would be interested in doing this. Teaching K through 12 students during neuroscience outreach events associated with Project Synapse. That sounds cool. I would want to do that. Mentoring. Wow, neuroscience. This was 2018. Is that German? Naturwissenschaften. I think that's what that says. Naturwissenschaften. That's so cool. Wissenschaften. No, Wissenschaften. Wissenschaften. Naturwissenschaften. Is that German? That's so cool. I just like saying it. Wow, she's written a lot of abstracts. Invited talks. Synopsis records CCLA. That's visual discrimination test. Dynamics, network tasks in the primary visual cortex. Experience decoding plasticity of network dynamics. Interesting. Collaborations. Fragile X research. Visual discrimination task in Fragile X syndrome patients. What are Fragile X syndrome patients? What is that? It's a genetic disorder. CDC. Okay. Fragile X syndrome is a genetic disorder caused by changes in a gene that scientists called the Fragile X mental retardation 1 FMR1 gene when it was first discovered. The FMR1 gene usually makes a protein called Fragile X, and they continue the mental retardation protein. FMRP is needed for normal brain development. People who have FX syndrome do not make this protein. People who have other affects both males and females. Females have mild symptoms. Signs that a child might have FX syndrome: developmental delays, not sitting, walking, or talking at the same time as other children the same age, learning disabilities for learning new skills, social and behavior problems such as not making eye contact, anxiety, trouble paying attention, hand flipping, acting, speaking without thinking, and being very active. Interesting. Fascinating. Very cool. And also very sad. There's no cure for the symptom, but there are different therapies that can be used to help children with the syndrome to learn skills. And they can take medication to help with behavioral issues. What interests you about this individual? I mean, I don't think that's all they're working on though. Oh, they are. So a lot of research with fMR1. Neural dynamics of in vitro cortical networks reflects experience temporal patterns. In vitro? So when the baby's still in the tummy? Or could that just be for anything? This is 2010. Let's see if we can find the article. Ooh, we can. Ooh, wait. What did they write? Circle mechanisms with sensory explanation and behavior. Cross-modal sensory input. Visual cortex. Network defect from temporal central. Polar sensory neurons. Ooh. Stimuli. Something X syndrome. My memory is horrible. Fragile. No, I don't like that. maybe. Okay. That is so cool. Zoom in there. He's so cool. Oh. He has a LinkedIn. Howard Hughes Medical Institute, postdoctoral fellow of the Life Sciences Research Foundation, Stanford University. Fascinating. PhD in genetics, California Institute of Technology, Brigham Young University, BS. That's wild. Alright, I'm gonna mute myself real quick just because I'm gonna help someone with something for stats, and I don't think they're gonna feel comfortable being on my live stream, so be back. Alright, I'm back. And I had messages. Aww, I'm not on TikTok anymore. Sorry, a friend of mine just messaged me and they were like, I made a TikTok and I don't know why. And they want to send me a TikTok, but I'm not on TikTok right now. And they're in a social media cleanse. I'll send it to them though. I'm pretty sure that was it. I don't remember. I don't remember my at. Okay, I should focus. I'm gonna focus. I don't know my at. I don't think it's on here either. Hold up. Like I said, accounts. I'm not logged in. What? Same shape? That is the weirdest thing ever. What? Oh. Oh yay. What is my at? And oh wow. When did I get that many followers? Okay. But like, Oh, okay, I guess, yeah, that makes sense. Okay. I'm gonna log out now. Where was I? I was up here. Yes, and microbiology. Oh, so he actually went straight to his PhD from a bachelor's. It's possible. PhD in genetics. And then postdoctoral fellow at Stanford. Institute. Okay, I'll be back again. Huh? Really? I don't know what to do then. How about now? Do you hear something now? Oh, interesting. Is it too loud? Do you want me to read? Well, the song just ended. Is it too loud? Not loud enough? I'm waiting. Can you hear me? Okay, I'm sorry. Google Scholar. I didn't really think Google Scholar was like a reputable place. But apparently it is. Applications. Course. Ontology.
Data clock.
Entomopathogenic. It smells like Christmas. He has a lot. Oh my god. Hematology. I wonder what kind of person dates a guy who studies nematodes. I mean, I would say the same thing if it was a woman. I'd be like, what kind of guy dates a woman who studies nematodes? I think that's so cool. I hope they have someone. Okay. I'm curious to know why he. Curious. Um, I know the answer, but I'm not. To control the. To control the. I'm searching. Managing. Immune modulation, tissue damage. I'm curious to know who this person was studying nematodes, was it raising cells for the creators that could have been studied for tissue purpose, could have been used for research purposes. I suppose your research is beneficial because they study the immune system. Okay, I need one more. He doesn't even have a CV. Like, he has a website. Mm, Root Park. Not enough information. Okay. I feel like they all say the same thing. Oh, I'll do music. Saavedra. Literally, that's it? Just that she has her PhD? Tell me about you, woman. Copy. Okay, where's her CV? What the heck? What? Watch if I've missed it. I still don't have my UCR card thingy. That isn't helpful. This wasn't helpful. Okay. I tried. Ooh. Her thingy just said, I'm looking for someone who can read or speak Spanish. And I'm like, that's me. Amalia. Amalia. Promises. Again, no CD. Ah, I found it. She got a BA from Pittsburgh College, Master's from Berkeley, PhD from Berkeley. Wow. Okay, just need the last one. Honestly, I just like to to intern for her. I'm not even interested in what she's studying, but I would just like to intern for her. Hi. How do I apply? I'm so done. Okay. No one's replied to them yet. Nature Vision Shopping. I just like saying it. Nature Vision Shopping. I don't know. Women's labor. Regions. That's all I have. Dude, she sounds so cool. Dominican Republic and Britain. Where do they disseminate their work? I have no idea. It doesn't say. Great Mechanisms. Oh. Ah. University of Chicago Press. Okay, that's funny. So this is from one of the works that she wrote. It's called Between Love and Money, Sex, Tourism, and Citizenship in Cuba and the Dominican Republic by Amalia Cabezas. She's like, gee, and I used to settle for a mint on my pillow. Comment from a recent traveler to Cuba on a sexual proposition that he received from a hotel chambermaid. Go Cuba, I guess. I want to read this. Can I read this? Oh. Full text. Nope. I would love to read this. Maybe I'll email it. I probably won't, but I would love to read it. I have to like sign in and stuff, so. She sounds so cool. Okay. What interests you as individual? Sustain. Did I really just write sustain? Sustain. done. Very cool. So now I'm going to turn this in. No one has made any Among Us memes? What? Wait, let me find one. Aww. Oh wait, she wanted to send me a video. Right. 8:35. Not a bad time to turn in an assignment. Like, who even turns in assignments on time? It's never been me. What is happening? I can win. I want to drink more coffee, but if I drink more coffee, I'm not going to go to bed. This one. Did I really? Wait, what? No. I think I did. No, it's in time. What? I'm so confused. Not like it makes much of a difference. Okay. Completed, yay! Okay, brain and behavior. Research and creating master disciplines, that is honors. What should I do? Stats is to Friday. Oh, yeah, I should probably do that. Finish writing that email as I was gonna write. Okay. Let's keep it short. Good morning, professor. This year is treating you well thus far. I'm currently an honor student trying to find a faculty mentor for my capstone project. Okay. The purpose of my research project is for studying the purpose of my research project is for studying the psychological impact of gendered microaggressions encountered in professional environments for Latinx women, and the effectiveness of their coping mechanisms. I hope this year has treated you well thus far. I am currently an honor student trying to find a faculty mentor for my capstone project. The purpose of my research project is for studying the psychological impact of gendered microaggressions encountered in professional environments for Latinx women and the effectiveness of their coping mechanisms. I have found that plenty of research has been done discussing the impact of racial microaggressions by white individuals toward black individuals, but current research has gapped applications to other cultural contexts. If you're interested in knowing more about the subject, I need to talk about their research. Capstone project. Megan Robbins, social interactions. That is literally, oh my god. Can you approach capturing pain, disclosure, and daily life? That's literally office hours Wednesdays. Tomorrow. Literally tomorrow. So for this professor, Meg Robbins, Megan Robbins. I need to make sure everybody's there. Good morning, Professor Robbins. It's still a respectful time. Good evening. Good evening, Professor Robbins. I hope this year has treated you well thus far. I'm currently an honor student trying to find seeking a faculty mentor for my custom project. What did you write? What did you say? What did you say? It is based on understanding I see you choose a space of understanding social interactions as well as well-being. I really believe connects to the particularly in expressive behaviors. I believe your research connects to what I would like to study, particularly in expressive behaviors in response to negative encounters. Negative, in the workplace. I really believe research connects to what I would like to study, particularly in expressive behaviors in response to negative or awkward situations in the workplace. The purpose of my research project is for studying to study the psychological impact of gender microaggressions encountered in the original environments for Latinx women and the effectiveness of their coping mechanisms. I have found plenty of researchers that don't discuss non-racial microaggressions for white individuals toward black individuals, but currently there is information in the cultural context. If this is something you might be interested in, I'd be happy to give you more information, but if not, that's okay. I don't know. Okay. Capstone. No one's really like. Who could I ask? Should I wait till tomorrow? I feel like I should just send it. Oh, I'm so scared. I know, I'm like doubting myself. Oh my god. Who should I talk to? Should I talk to Stephanie? I should be independent. I'm freaking out. I think I have a draft. I'm gonna send it. Yay! Oh, cool. Oh, wait. Ooh. Oh, that's so much.
romantic talk.
Yeah, Ambassador Hangout. That's the Ambassador Hangout. It's not required for me. I literally don't know why. Like, I'm like so confused. Ambassador Hangouts are only for first years. I'm not a first year, I'm a transfer. So I'm like, what the? I'm looking for an email because I remember that once the university honors thingy was just like, make sure to put this in your signature. And I'm like, huh? Okay. And then I never did it because I forgot about it. I really feel like they sent that email as a mistake. I really think so. Like, they're taking their sweet time to reply to my email, to my appeal. I'm pretty sure they made a mistake. I think this is okay. I really think it's okay. I'm so scared. It's 9. It's 9. Oh my god. I sent it. There's no undoing. Well, technically, yes, I still have a few more seconds, but oh. I'm so nervous. god. I'm so nervous. She's so pretty. I'm just like, damn. My leg. She's literally so pretty. I'm just like, how? Okay. Look at her. So pretty. Okay. And then I have to figure out how to submit. I wonder if we'll go over that tomorrow. Should I be looking at other faculties? Other faculty peeps? I mean, she still has room. Oh, I didn't know Dita was. Oh. Prejudice integrate processing. That could be a potential. Implicit bias, attitude formation, change persuasion. Maybe. Think about it. What's his name? Jimmy Camacucci. If I get rejected, I'll be so sad. So sad. Very sad. Socialization, cultural influence on human development. I mean, that's a stretch, but it could be. Just and there's one more. He said no. Can you take a university honors? He's like, no. That's a good question. I am so freaking out. Okay, I feel like I did a lot for this class today. I feel 9. Wow. Okay. She's so pretty. What else was I gonna do? I'm gonna do it tomorrow. Brain lecture on user discussion. Yeah, we ended up going through all of this. Cells are polarized. They are monometric on the inside. And properties of membrane potential. Speed. I'm lowering the volume because my family sleeps very early. I just realized it's 9:30. So right now I am working on my active recall questions for my learning behavior class. Because this class, the final exam is going to be cumulative, so that's going to be killer. So I'm trying to set up questions after each lecture and discussion after I've gone through my notes. So that's what I'm doing right now. That's literally the question. Okay. Oh, okay. Saltatory conduction. I think at this point I was dying. And this is kind of where he ended. He kind of just talked about these really quickly, but he didn't really go into detail. So I don't really understand saltatory conduction or these thingies. Yeah, we kind of finished here. The diversity, and then this, all of this is probably be for tomorrow. We'll see. Yeah, a lot of stuff, since I had reviewed it the day before, during the weekend, I understood a lot of what he was saying. So that was good. So I already did some questions here. And alright, so let's do some active recall. I study this one. I'm trying to figure out like how to review. Ooh. 1, 2. Ooh, okay. So I'm gonna make sure to do that later. Okay, review 2. Levels of analysis. What is the process behind our behavior? Oh god, I remember going to this. What is the process behind our behavior? Behavior, I know, has to do with external, internal processing and integration. Yes, I remember integration. And then how that comes out in our behavior. What is the process behind our behavior? So obviously there is process of processing our internal and external stimuli, and then integrating that like in our brain processes, and then our response to all of that stimuli is the outcome of our behavior. Okay, intake, I kind of said that. And actions based on this processing. Yes! OMG. Okay, I'm gonna give myself a blue because I doubted myself. What is the neocortex? The neocortex, I will never forget what the neocortex is. So neocortex is basically, let's say you have a brain, and then just imagine an orange. The neocortex is the skin of that orange, the rind of the orange. Um, it's a region of the brain, the external region of the entire brain is the neocortex. External region. Oh my god, I said the right word. Okay, that deserves a green. That is done. What are neurons? Neurons receive and transmit information over in the brain. How'd I do? Receive information and transmit it to other cells. Yay! I got that right. I feel good. I'm gonna give myself a blue, just so that I test myself again. Oh, two. Okay. Oh my god, don't show me anything. What are glia? Okay, there's different kinds of glia, but glial cells, they help with a lot of different processes. There's a lot more of them than there are neurons, which is interesting. Glia can continue to duplicate even as adults, whereas neurons cannot. Glia, the different kinds is astrocytes, oligodendrocytes, Schwann cells, microglia, and then there's one more that we didn't really talk about in the lecture, but it's mentioned in the textbook, and that I cannot remember now. What are the glia? They all have different processes. Most of them are basically just helping neurons. They're like support systems, support structures that help neurons do their job. That's kind of. Okay, I didn't write that in there, but that's what a glia is. Okay, I'm gonna put you as yellow because I took too long. All right, cells of the nervous system, neurons and glia. How do we count the number of neurons in the brain? So it's a process of homogenization. It's very difficult to count the number of neurons in the brain because, first of all, everyone has a varying amount of neurons in their brain. Like no one has the exact same amount. And then different regions of the brain have more neurons than other regions of the brain. And then they're not very like uniform. There's no uniformity basically. Like they're just scattered all over the place, and some places are more concentrated than others, and so it's really hard to count them. So how do we count them? Basically, scientists pick specific areas of the brain, take a little sample or whatever. I think from like a dead brain. I'm not sure. Pretty sure not from the live brain because that would be painful and unethical. So basically, they will put it in a they put it in like a they will suspend it in a serum like a water thingy. They'll put them in that, and then they had to basically homogenize them. So just being there, like they're not going to be uniform. So they try to create uniformity by homogenizing that sample of the cells, and then they stain the cells. That's what I always forget. They stain the cells in order to count them. And there's like specific markers that they use to track which of the stuff in the tube. Which of the stuff in the tube? That should be a quote. Which of the stuff in the tube? Which of the stuff in the tube? I'm never gonna forget that. Is an actual neuron, because there might be other stuff in there too. I can explain it, alright. Centrifugate. Sure. And then they stain the cells. Yeah. Okay. How many neurons and glia are in the brain? So there are different numbers. I think I remember going through these. I don't think I actually have to remember these, but I kind of just want to know them. And I realize that I'm not in frame. Wait, am I in frame? I'm not in frame. Oh, hi. Oh, they left. My bad. Oh, wait. I'm gonna give myself a blue. And then how many neurons are in glia are in the brain? I'm going to do this right now. I remember these numbers. So there are like really crazy numbers. There are the textbook numbers, the widely accepted numbers, and then what it probably actually is. What it probably actually is probably like 85 billion neurons or 85 billion glia. I think the textbook answer is I think there's 10 billion neurons. No, yeah, 1 billion neurons, and then I think there's like 10 trillion. It could be 10 billion neurons and then like 10 trillion glia, I think is the number, or 100 trillion. Yeah, it gets wild. And then the widely accepted answer is, I believe it's 1 billion neurons and then 1 trillion glia. Oh, 100 billion neurons and then 1 trillion glia. Okay, I was close. Neurons. I did it. 1 trillion, 10 trillion. Widely accepted. I don't think I'm actually going to need to know that, but I want to know. Textbooks, 100 billion to 10 trillion. 100 billion, 10 trillion. Textbook, 100 billion, 10 trillion. And then the widely accepted is 1 trillion to 10 trillion. Yes, got it. I'm going to give myself an orange on that one. That one's hard. Types of glial cells. What are the general properties of glia? Oh, it's those 3 bullet points that I never know. They are like a support system for the neurons. Support structures. They don't transmit messages. Kind of help with the immune response, and I don't know what else. What was it? I'm gonna give myself an orange because I got one right. General properties of glia. They divide as adults. Non-conducting. That means they don't have charges. Which is important because there's a lot of charges going on inside the neuron. And then they've I'm gonna work in supportive roles and metabolic roles. Okay. Like I said, I was giving myself an orange for that. How do astrocytes interact with neurons? Okay. Neurons. Okay. How long have I been streaming? It's gonna be 3 hours? Oh my god. Okay. I'm crazy, huh? For studying for like 3 hours? I know. Oh. Cells of the nervous system. How many neurons and glia are in the human brain? Where was I? Right. How do astrocytes interact with neurons? So astrocytes, they do a lot of metabolic functions. Astrocytes actually attach themselves to capillaries, blood vessels, and cell walls. Not necessarily like as an anchor or anything, but they just attach to them. What else do they do? I don't know. That's a lot of stuff. They attach to neurons, synapses, blood vessels, and capillaries. And they attach. Neurons, synapses, blood vessels. Remove waste material created when neurons die. That's microglia too. Control the amount of blood flow to each brain area. Huh. Scaffolding for neurons, especially during development. Astrocytes. Uptake, release of chemicals, including neurotransmitters. Synchronize the activity of axons. Modulate synaptic transmission. Okay, astrocytes. There's 6 things. Okay, six. Okay. They attach to capillaries, synapses, blood vessels, neurons. They attach themselves, surround processes. Okay. They pick up waste materials from dead neurons. They help with the immune response. No. Did I say that? I think they no, I don't think they do. Well, we'll come back to that. They modulate synaptic transmission. They receive, or no. It's kind of like they're a port for neurotransmitters. A port, so that's uptake, release. And you think that a port, a ship landing, a boat, a ship, a vessel, a ship vessel, boat vessel. Uptake, release, import, export of chemicals, including neurotransmitters. That's such an important one. Control the amount of blood flow to various brain areas. Control the amount of blood flow to. Okay. These 6. So what are the properties of astrocytes, right? How do astrocytes interact with neurons? So in development, I'm going to move these. Right. Okay, I move them so they make sense to me. So astrocytes in development, they act as scaffolding neurons, things to hold on to, right? Okay, and so that actually also makes them a point for uptake and release of chemicals, including neurotransmitters, because they act as a port for those things. They control blood flow to various areas of the brain. They also pick up waste materials for when neurons die. What's it called? They attach themselves to, kind of easy, oligodendrocytes in the central nervous system, the CNS, primarily in the spinal cord and in the brain. Whereas the Schwann cells are primarily found in the peripheral nervous system, the PNS, and so they have different names. They both have the same function, they just myelinate the axon in different areas of the body, because some things can enter the spinal cord brain areas, and those can probably enter, but stuff in the penis probably can't enter the CNS. So that's what differentiates them. They both myelinate though. And what is the role in the neuron? They myelinate the axon. The axon basically allows the action potential to travel down the axon quicker. It works as like a buffer almost, so that the axon doesn't go crazy. Yes, yes. Uptake release of chemicals, including neurotransmitters, modulate synaptic transmission. I mean, I basically said modulate synaptic transmission. I just didn't say it in those words. I just said they myelinate the axon, which allows the synapse, the action potential to roll down the axon. I'm gonna give myself. There's already a yellow. I think I will give myself a blue. What are the properties of microglia? Microglia is actually really cool. Microglia, bundles of microglia are actually also called white matter. They work as like an immune response due to inflammation in the brain. So they fight off that inflammation. They also work as janitors, kind of like the astrocytes, in that they pick up waste materials as well. And then what else do they do? Microglia. I think that's kind of it. Let's see. Oh yeah, there's this whole thing about the cytokines. I did talk about cleaning up debris in the brain. Uptake release, modulate synaptic transmission. I feel like they all do that. Modulate synaptic transmission and immune response to the brain. But I don't mention it. So maybe I should put it here. I'm gonna add it here. Just because I feel like they all contribute to that. I'm gonna give myself a blue for microglia. And then how do bad glia behave? So bad glia, okay, so this has to do with regeneration and the fact that regeneration doesn't really occur in the central nervous system. So when people have like injuries to their spinal cord or like areas that are like important to the nervous system functioning, that means like being able to move your arms, being able to move your legs, that kind of thing, your face. Like all of those functions reside within the central nervous system. So regeneration doesn't occur within the CNS. So nerves that are damaged that belong to the CNS, the central nervous system, those nerves can't regenerate. They can't fix themselves or like create new ones to fix the problem, as compared to the nerves in the peripheral nervous system, which is like things basically I would say closer to the skin in our external body systems. So like if you were to damage the nerves in your arms or in your legs, kind of that aren't reliant on the CNS, like those are able to regenerate. So those can fix themselves, create new ones. And so that's why the glial cells in the CNS are considered bad glia, because they actually inhibit that regeneration process. And so there's research right now that's actually trying to see how nerves in the PNS, the peripheral nerve system, can be used in the central nervous system to basically act as bridges and allow the CNS nerves to regenerate. I'm pretty sure I got that. Of course, I said a lot more. Molecules expressed in the CNS don't regenerate. Yep. Inhibit glia. Glia inhibit that regeneration. And they're trying to improve the CNS regeneration by using PNS tissue as a bridge. Block molecules or promote growth. I'm giving myself a blue. And hopefully next time I remember it and it turns into a green. So my goal is to have all these become green. All right, plasma membrane. What kind of activity takes place in the plasma membrane? A lot. Okay, so the plasma membrane is a lipid bilayer. And so lipids So you have phospholipids within the plasma membrane. You have the heads, and then you have the tails of the phospholipid. The heads are hydrophobic. No, hydrophilic. I forget which. The heads are hydrophobic. The heads are hydrophobic because they keep out the extracellular fluid. And then the tails inside are hydrophilic, which keeps fluid inside, right? Okay. I will see if I got that right. What kind of activity takes place in the plasma membrane? So the plasma membrane basically acts like as a gateway, allowing certain things in, allowing certain things out. And this is just related to the blood-brain barrier that we'll talk about later on, because the membranes in other areas of the body kind of have like gaps in between, and they kind of let things in and out. But the blood-brain barrier is like super locked in place like this, and really just doesn't like to let things in more than it lets things out. So I think that's a good lipid bilayers that allow separation from intracellular, extracellular environments. Yeah, proteins are embedded in the layers. Some things allow things to go through the membrane, acting as channels, and some as receptors. I didn't really say all that, so I'm gonna keep it blue. But yeah, I didn't even talk about like the the heads and tails, hydrophilic, hydrophobic stuff that I kind of already know, so it's fine. The axon. What is contained within the cell body? The nucleus and the DNA that is inside the cell body. Nucleus and the cytoplasm. Sure. Cytoplasm. Nucleus, DNA, cytoplasm. Alright, I'm gonna give myself a blue. Okay, so I didn't say cytoplasm. What is another name for cell body and its plural form? The cell body is also known as a soma, and its plural form is somata. Somata. Yes, I'm gonna make you green. What are the properties of an axon? I'm assuming all of its little parts. So we have the axon, we have the cell body here, and then coming out of the cell body are the dendrites, and then you have the axon hillock, and then you have the axon. I don't know what this is called. I'm guessing that's the axon. And then you have the synapse terminals, or the dendrite terminals. I think they're synapse terminals. And then you have the myelin. Do I have to go through each? I think this is the one that wants me to go through each. What are the properties of an axon? Okay. There's only one axon per neuron, and that's all I know. Axons are adapted for more distance travel, transfer output and flow. The axon has a set of action potential. Okay. Let me see this. Axon has three things. The axon breathes. Okay. I feel so dumb. Damn, that's already 3 hours of studying. It's the coffee. I haven't talked to y'all, but no one ever likes to talk back, so I'm just like, okay, so I'll just keep talking to myself. Can you even hear me? I don't think you can hear me. I don't know. Anyway. I still have so much to study. Have I even prepped for tomorrow's lecture? I haven't. Okay. So this live has been 3 hours long, and now I'm just talking to myself, but I'm actually talking to you. I should look at the camera, huh? No, it's awkward. So yeah, I have accomplished so much today. There's a lot that I studied, and I just want to say thank you to everyone who was able to study with me. What's it called? I'm excited to keep this up and keep studying as much as I'm studying and really using my time wisely. And I hope this is inspiring for y'all, so y'all feel motivated to study too. Honestly, like if you don't feel motivated, coffee kind of makes your mind go you. So drink some coffee or some tea, or eat sour candies like I usually eat. I don't know, whatever keeps you awake. And then whenever you're studying, like everything's new, so it should feel kind of exciting to learn something new. Don't be afraid to look like an idiot. I mean, I'm pretty sure I look like a weirdo in my live stream because I'm like getting excited over all these little small things. But yeah, that's kind of learning. You have to be excited about it, to make it interesting, to make it fun. Don't care about what other people think, and if they think that something is boring or something is like, why are you getting so excited about that? Like, don't let, like, bro, it is, you know? But anyway, okay, that's enough of my little speech. Good night, and until next time. Bye.